Hi and welcome back. We're going to continue our objectives 30 to 33 where we were talking about muscle contraction and movement. We're going to give a little bit more specifics on types of contractions, types of muscle fibers, and the actual brain areas and peripheral nervous system areas that are involved in the action of triggering a movement. So we're just going to continue on a bit. One of the things that you do cover well in lab, but I think that aren't really a great part of my notes I wanted to add in here, and that's what a motor unit is. A motor unit is all of the muscle fibers, muscle cells, that are supplied by a single neuron. And there are many motor units per muscle. And each one contracts as a group. So I wanted to show this to you. So you may have a nerve, and that nerve will contain many axons from many neurons. And once that nerve enters a muscle cell, you'll see it branch. And all of the individual axons you see will supply individual cells. So this one axon here supplies this cell, and this cell. So you can see the neuromuscular junctions of each one. So this is a single motor unit, all the fibers that are controlled by a single axon. Whereas over here we have a separate neuron, separate axons, and separate cells. So inside a muscle, you may have many motor units, and the motor units are controlled by a single axon. So when this axon triggers the cell, both this and this cell would contract at the same time with the same degree because of the same action potential and synaptic transmission. If you want to control the amount of force that a muscle produces, you can recruit more motor units. If you recruit more motor units, then the entire muscle can contract with greater force. That's called recruitment. So if you pick up a feather, you may use 1 or 2 motor units, for example, maybe 10 cells. This is an exaggeration. But if you pick up a 10-pound weight, you might need to use 20 motor units or 50 cells to create the force that you need to pick that up. And so to create various forces, or even to create various types of movement, you may recruit different amounts of motor units. So let's get back to the notes because it's going to kind of play in. We're going to talk about what a muscle twitch is. A muscle twitch is a brief contraction of all the muscle fibers in a single motor unit, and that's in response to a single action potential from a motor neuron. So one motor neuron tells one motor unit to contract, and it does. It's the simplest type of recordable muscle contraction, and there are components of a muscle twitch that we can identify. So the very first one is called the latent period. So you see on this diagram how at 0 milliseconds we get our stimulus, but we don't start doing anything, contracting until maybe, let's say 1 or 2 milliseconds. Why is that there? That is the latent period. It's the period between the stimulus and when contraction begins, between the stimulus and when contraction begins. Why is it there? Well, remember we learned about excitation-contraction coupling? It takes time for the action potential to be conducted along the length of the sarcolemma. It takes time for that action potential to dip into the T-tubules, to trigger the sarcoplasmic reticulum, to release the calcium, for the calcium to diffuse among the sarcomeres and finally bind to the troponin. So we don't see contraction until that myosin head first makes contact with the actin active site, and that's when contraction takes place. Now all of that only took about 1 or 2 milliseconds, but that still takes time that we can record. So the contraction period is simply the muscle contracting, and the relaxation period is a muscle returning back to its uncontracted state, so repolarizing. But one of the things I wanted to mention about that repolarization is that during this relaxation, and even during contraction, we have what are called refractory periods. So soon as we depolarize that cell, there is a period of time when a cell maybe could not contract again, and that's a refractory period. But there are different kinds of refractory periods, so let me be more clear. An absolute refractory period extends from the point of threshold stimulus until depolarization is 1/3 complete. During this time, this upshoot, we cannot stimulate a cell to contract again, even if you try. The relative refractory period is from the end of the absolute to where depolarization can begin again. So this whole repolarization phase is the relative refractory period, and if the action potential or the stimulus is strong enough, we may be able to contract again. Now, why are those there? So I have a little question for you here. Skeletal muscle has a short refractory period, but cardiac muscle has a long one. Why do you think that is? Think about how they contract. If you're trying to show off your bicep brachii, your muscles, go ahead and show that off. No one's watching you. Contract it, feel how hard it is. Contract it really, really, really heavy. Keep contracting it. Don't let go. Keep contracting it. That's called a tonic contraction. You are sending a constant stream of action potentials to that muscle so that you can contract continually. That's skeletal muscle. Very short refractory periods. Go ahead and just stimulate, stimulate, stimulate. Now imagine your heart. Do you want your heart to be able to do that? Do you want your heart to be able to contract as a solid muscle for a sustained period of time? No, because it's not acting as a pump. We actually need our heart to be able to contract and relax. Contract and relax, so that it can act like a pump to allow the blood to enter and then be forced out. So we have long refractory periods to prevent a tonic contraction in our heart, but we have short refractory periods to allow it in our skeletal muscles. I hope that makes sense. Let's talk about those tonic contractions, or what we might call tetanus. So in skeletal muscle, if a muscle receives repeated stimuli at a really rapid rate, it can't relax completely between contractions. So the tension that's actually achieved under these conditions is even greater than the tension or force for a single muscle contraction or a single muscle twitch. That's called wave summation, when it actually increases, as you see, step by step by step, it increases in force or tension in that contraction. So a single twitch has low tension. We can increase if we continue to contract. And if we continually contract, look at the force now, very high. You can see there's a difference between incomplete tetanus and complete. With incomplete tetanus, very much like wave summation, we have little periods of relaxation in between those stimuli, but we do still increase in the force of that contraction. Whereas tetanus, we have a very fast rate of stimulation, no relaxation time between that stimulation, and so we have a continuous contraction of a muscle at its greatest force. Well, how does that happen? Down here it says increased force results from calcium being released in the second stimulus and the third, while the calcium ions are still present from the first. So we release calcium to get contraction, and before all that calcium can be retaken back up into the sarcoplasmic reticulum, we stimulate again. And that second stimulation provides more calcium. And before that calcium has a chance to be taken back up, we stimulate again and give us more calcium. So that's actually the beauty of, if you're going to go out and run a sprint, to warm up those muscles a little bit. Get the calcium flowing so that you can actually get more force than you would have if you ran cold. I owe my track coach an apology. I didn't understand that back then. This next slide is just a reminder. You're going to actually have a lab, or maybe you already did, on electromyography or the EMG. And you're going to be testing recruitment. You're going to be testing how muscles can increase in their force or tension, and also kind of comparing dominant to non-dominant arms. So you'll actually get to see those kind of in action with you when you print off your simulation of the EMG. So I want to spend most of our time on where the energy comes from in order to contract, and that starts with objective 32. Now this slide is super busy, and I get that, but it's really, really, really great for explaining all the different ways that we get energy. So we're going to kind of take it piece by piece. We'll start over here on the left, where we have a molecule called creatine, and creatine can help us to make the ATP we need for energy. We'll move into how glucose is then used for energy in a glycolytic cycle. And then we'll move into glucose using for energy and how it moves into what we call oxidative phosphorylation, okay, or oxidative, an aerobic mechanism for producing energy. So we're going to take them piece by piece. We'll come back to this picture as often as possible. And this is also in your notes because I found it so important for helping us to understand all these aspects of contracting and energy. One of the first things we have to kind of remember is energy, what the energy is. ATP, adenosine triphosphate, is our energy source for muscle contraction. And it is how muscles are able to contract. We know that we need it for cross-bridge cycling. We know we need it to reuptake the calcium back into the sarcoplasmic reticulum. So it's absolutely essential. We cannot move without that ATP. So we have to generate it. Well, how much ATP do you actually have on hand? You only have actually about 5 to 6 seconds worth of ATP on hand within a muscle to contract. So not very long. What's happening is this adenosine triphosphate molecule has 1, 2, 3 phosphate groups. And when you cleave off the last phosphate group, it becomes adenosine diphosphate plus a phosphate group, but a lot of energy that can be used to power contraction. And so we actually need all this ATP to run our contraction scenario. So if we only have 5 to 6 seconds on hand, where does the rest come from? I like this slide because it can tell us where the rest comes from. 5 to 6 seconds is how much ATP you have on hand to get started, to get moving. The next maybe 10-ish seconds comes from a molecule called creatine phosphate. We get creatine phosphate, we're going to talk about that, can break down to release, to create ATP, but not, again, very long. If you really want to work out for a length of time or move for a length of time, we have to get sugar involved. Glycogen is how we store sugar. Glycogen can be broken down into individual sugar molecules or glucose molecules, and that glucose can be converted to produce ATP. But anaerobically, maybe only for about 30 to 40 seconds. Anaerobically means you're not providing oxygen. If you provide oxygen and sugar, you could exercise for hours. We can produce ATP almost with no limit. And that's what I want to show us. How are the different pathways given to us? The creatine phosphate, the glycolytic pathway, and the oxidative pathway. Let's talk about creatine phosphate first. So what you're looking at, here's a muscle cell. Here's your blood supply. Creatine phosphate is within the muscle. And this molecule is kind of a high energy molecule. And if you break it apart into creatine phosphate, you break it apart into creatine and phosphate, that provides enough energy for us to create what's called phosphorylate and create ATP. We just add that ADP and the phosphate together to make ATP, but you need energy to do that. So creatine phosphate provides the energy to phosphorylate and make ATP. But again, 10 to 15 seconds, that's about all you have of your stored creatine phosphate to produce the ATP you need to run a muscle. If you really want to have a muscle be able to perform, contract for longer periods of time, that's when you have to get glucose involved. You can take glucose from the bloodstream, you can take stored glycogen and break it down into glucose, but that's when you're going to undergo two pathways. You're going to break that glucose down either by glycolysis or by oxidative phosphorylation. And that's the best way to produce ATP that we have and run that muscle for a long period of time. Let's talk about glucose then. Glucose is used for prolonged exercise, and that's anywhere between 30 seconds to many hours. Glucose, we know, is stored as glycogen, but during exercise, you can break that glycogen molecule down into individual glucose molecules, and then glucose has two choices. Here, it could undergo what we call glycolysis, or glucose could undergo what we call oxidative phosphorylation. We're going to talk about glycolysis first. In glycolysis, which does take place in the cytoplasm of your muscle cell, glycolysis is the breakdown of glucose into what's called pyruvic acid and energy. So when you break down sugar, which is actually 6 carbons in a row, into pyruvic acid, that's a 3-carbon molecule, you break it down into 2 pyruvic acids and 2 ATP molecules. So you get a little bit of ATP from this process. But most importantly, what I need you to see here is you're not providing oxygen. This can take place without oxygen. Once you have that pyruvic acid, you either have to provide it with oxygen here, so it can enter into oxidative phosphorylation. But if you don't continue to provide your cell with oxygen, that's when that pyruvic acid can become lactic acid, and that's when you start to have muscle pain. We'll talk about that soon. This is just a nice picture. I like how they kind of depict glucose. You see glucose here is a 6-carbon sugar. We're going to break it down to produce 2 molecules of ATP, and that's going to produce 2 of these pyruvic acid 3-carbon molecules. And so long as you have oxygen present, that pyruvic acid can enter into the next step, which is oxidative phosphorylation. This glycolytic pathway is called anaerobic because it does not require oxygen. It's pretty quick energy, but it doesn't provide very much. It's a fast energy, but maybe only for 30 seconds. If you really want to supply muscle with hours of energy, you have to undergo oxidative phosphorylation, entering the Krebs cycle, what we call chemiosmosis. And that's what we're going to talk about now. Okay. So here's just that one diagram. We see the glucose being broken down. Oxygen does not need to be present into pyruvic acid. But if we want to take it a step further, we're going to enter our mitochondria with that pyruvic acid. We're going to have oxygen. But then we can actually break that pyruvic acid from the sugar into 36 ATP molecules. And then our end products are only carbon dioxide and water. So it says the pyruvic acid enters the mitochondria. Here it is completely broken down into carbon dioxide and water and a high output of ATP. This is called aerobic respiration or cellular respiration. It's also called oxidative phosphorylation. Lots of names. But the key is you have to provide oxygen and the pyruvic acid. This is really different than glycolytic because glycolytic provided 30 seconds. This aerobic pathway, oxidative pathway, can provide hours of energy. So that's a difference in some of your muscles. Some of your muscles are glycolytic muscles, and some of your muscles are oxidative muscles. So some of your muscles can actually work longer, while others literally aren't designed to do that. Well, that's great if we have oxygen present, but what if oxygen isn't present? So for example, again, we break glucose down into pyruvate. If oxygen is present, we enter the mitochondria, enter the Krebs cycle, and ultimately we're going to produce about 36 ATP to keep you running. But if you produce pyruvate from the glycolytic cycle, but then there's no oxygen to keep going, the end product is different. It's lactic acid or lactate. Lactic acid is an end product when you try to break pyruvic acid down without oxygen present. Lactic acid can stay inside that muscle cell, and it can also diffuse into your bloodstream. And this is what contributes to muscle fatigue, it contributes to pain, it contributes to breathing heavy. Okay. We know that oxidative phosphorylation can take place if oxygen is present, but if it isn't, then we may enter into, again, fatigue and oxygen debt. Oxygen debt is the amount of oxygen needed to be taken into the system after exercise to do three things: to metabolize or break down your lactic acid, to literally remove it from your muscle cell, to restore your creatine phosphate supplies, and also put that oxygen back into your myoglobin so that you can work harder, longer. If you are working out and you begin to breathe heavy, you have surpassed your myoglobin stores. You're in oxygen debt, and you're now providing what oxygen you can to those tired muscles. And again, that's what I was saying before. If you begin a workout program and maybe you're on your treadmill for 5 minutes and you start to breathe heavy, that's because your myoglobin stores are very, very limited. But as you continue to increase your cardiovascular health, you build more myoglobin stores, you can exercise a lot longer before you start to breathe heavy. And that's because your oxygen debt level has not been reached. And so you don't have to compensate through breathing. You have lots of oxygen already in your muscle cells. But what happens if we don't have enough, or you've reached your capacity? Lactic acid can build up, and what it does is as it diffuses into the bloodstream, it actually will trigger your respiratory center of the brain to begin that rapid deep breathing to replace the oxygen, and also to exchange the lactic acid in your muscle cells for oxygen. That's what helps to pull it out. And that's also why you should kind of stretch the next day. It helps to, instead of sitting on the couch because your muscles are sore, it helps to actually switch the lactic acid, kind of exchange the lactic acid for your oxygen, and it's going to make the pain go away faster. The more exercise to which you do, the higher your oxygen use, more efficient oxygen use, and what we call the higher your anaerobic threshold, the tipping point when you have to start really breathing heavy and you're entering into only glycolysis. And of course, the lower your oxygen debt will be. So the more you exercise, the more your aerobic respiration, the more you decrease your oxygen debt. Ultimately, muscle fatigue can happen, and that's literally the inability of the muscle to maintain its strength of contraction after a prolonged use, and that's due to lack of energy and lack of oxygen. What happens is the muscle, when it's repeatedly stimulated and produces a high amount of lactic acid and depletes its ATP stores, the muscle cell, due to that lowered pH, could actually stop working. No ATP, stop working, and low stores of myoglobin, low stores of oxygen, stop working. If you exercise long enough, you may actually not be able to lift that weight one more time. And that's what we call fatigue. You know, there are different kinds of muscle fibers as well. You've heard, I'm sure, a long time about slow twitch and fast twitch fibers. Well, what does that really mean? When you talk about a slow fiber or a slow twitch fiber, sometimes those are called type 1 fibers, it just means they have a lower ATPase activity. It means that ATP and myosin cross-bridge cycling happens at a much lower, slower pace, about 4 times slower than a muscle that's actually faster. The good thing about a slow twitch fiber or type 1 fibers is they have a great oxidative capacity though. They are the best fibers if you really want to think about it for long distance running. And so they're going to have really great myoglobin stores. These will be kind of like your red muscles, if you will. Now, fast twitch fibers, also called type 2 fibers, they contain a high ATPase activity. That means they just split ATP really fast so they can form cross bridges really fast and cycle and pull on those sarcomeres really fast, 4 times faster. But there are actually 2 kinds of fast fibers. There are fast fibers that can go a long time, and there are fast fibers that run out of energy really quick. So fast oxidative just means they have more mitochondria, so they have better capacity for oxidative phosphorylation, the Krebs cycle, making lots of ATP with lots of oxygen. So there's going to be better blood flow to these types of muscles. They're going to be more myoglobin, so let's call this kind of pink muscle. And then the fast glycolytic, very few mitochondria. They do have better glycogen stores, but very few mitochondria, very little myoglobin. They're actually kind of white fibers, if you will. So if you think about red meat versus, or light meat versus dark meat, chicken breast versus chicken leg, that's what we're talking about here. Chicken breast would be white meat, probably fast glycolytic maybe. The red meat would be the slow, and it would be the red meat, more oxygen supply, more blood supply, and even a little fatty to provide that energy. I really like this picture because it just does take our slow twitch and our fast twitch down to the most basic ideas. That slow, really good use of oxygen, whereas fast, not so much. They don't burn oxygen to create energy. It's more glycolytic pathways. Delayed muscle firing for the slow, but really fast muscle firing for the fast twitch. Slow twitch don't tire as easily, and they're better suited for like your marathon running, whereas fast twitch are better for you sprinters out there. This is also a really nice slide if you have time to look at it later. It just gives you even more detail with the slow. This is going to be your red muscle, fast oxidative, more of your pinkish, and then the fast glycolytic, that's your white meat. Okay. Metabolism, these are oxidative, mostly oxidative, and all glycolytic. Not very powerful and a little slow, intermediate, and this is your larger muscles, the most powerful, and your sprinting muscles. And again, capillary blood supply, great. Capillary blood supply, not as much. So to finish up this talk, we're going to talk about objective 33, which is now what areas of the brain are controlling our ability to move our skeletal muscles. We know skeletal muscles are under voluntary control. We know they have to be activated by a neuron, the axon terminal, and a neurotransmitter, acetylcholine. But what areas of the brain are sending those messages? And so that's what we're going to talk about right now, movement. We know that movement involves really complicated processes of nerves, muscles, bones, all working together. Think about reaching out for your coffee cup, picking it up and carrying it to your face. I'm going to do that. Yum, coffee. What did I just do there? I flexed my forearm. I extended my forearm. My coffee cup was heavy because I haven't drank it. What was my hand doing to grasp? What did my brain initiate? Was my elbow, my shoulder, my trunk even involved? All of these things have to be considered, and areas of the CNS, your central nervous system, and PNS have to be considered as well. When we talk about your central nervous system, basically your brain and spinal cord, we want to talk about the major areas, what we call the sensory motor cortex, to control all of those skeletal muscle actions. The sensory motor cortex is every cortical area that has a role in movement of skeletal muscle. And up here you can see, for example, some major cortical areas. Cortex is the outer layer of the brain. This cortex has the primary motor area here in pink. It has the premotor area here in blue. It has Broca's area here. These are just a few of the areas, but this primary motor area, that has the major control. If you want to reach out for that coffee cup again, the primary motor area was the primary area to help you to grasp. Premotor is more like driving a stick shift car. It's skeletal muscle action, but almost of a reflexive nature, kind of learned motor skills that you don't have to think about anymore. Procedural memory. We also have Broca's area, which are all the motor units or muscle units that are involved in speech. So moving your lips, your cheeks, your tongue, your palate, your glottis, everything that you're doing, or maybe I'm doing right now, to control speech. Let's focus a bit on that primary motor area though. I love this picture because you can see right here, this primary motor area is at the most posterior aspect of that frontal lobe. And there is a gyre here. It's called the precentral gyre. That precentral gyre on the left and the right is our primary motor area, primary motor cortex. And it's really going to be the major control of your skeletal muscle movement for your entire body, mapped out on this precentral gyre. Also remember that if you grab your coffee cup with your left hand, that would be the right precentral gyre that initiated that movement. The primary motor area uses really some of the largest neurons, and those are called pyramidal cells. And they allow us to consciously control the movements of our skeletal muscle. They have this, for example, this would be the motor cell and the axon. Very long axons. They usually do not synapse on their way down, not until you get to that anterior horn of the spinal cord. You notice there's no synapse and they cross over, decussate, or have contralateral innervation. They cross over to the other side of the brain. So that's when I said if you grab your coffee cup with your left hand, then that would be the right frontal lobe primary motor cortex that initiated that. There are also areas. Now this is primary motor area is every skeletal muscle action you have, but it's kind of in its basic form, rudimentary. We also have lots of other subcortical areas that can help us to fine-tune that motion. Make it smooth, make it coordinated, start it, stop it, control force. And so that's what we're going to look at now are some of the other areas, not the cortex, but areas below the cortex that might really help us to control our movement. The first area I want to mention is called the basal nuclei, which is also called the basal ganglia, which is this area of gray matter deep to the cortex. The basal nuclei actually receives input from your primary motor cortex. It also receives input from sensory areas, and it helps to influence movement by directing that primary motor cortex. What it does is it helps us to sort, stop, monitor movements. It can regulate the intensity of our movement, and it inhibits movement when we don't want to move. If you impair the basal nuclei, you will have poor posture, lack of muscle tone. You'll have involuntary movements, like moving when you're at rest. If your arm is sitting on the desk right now and it just had a continual tremor, that resting tremor is a product of a basal nucleus that is becoming diseased. You also have abnormally slow movements. That's called bradykinesia. And this is most seen in Parkinson's disease, where the basal ganglia, the basal nuclei, is starting to become atrophied. With Parkinson's disease, that main characteristic of the resting tremor is what you're going to look for, but possibly very slow movement or no movement, bradykinesia or hypokinesia, very rigid extremities and neck. You will lose facial expression capabilities. And the problem is that we are losing the neurons that produce dopamine in this basal nuclei. There's another area that's really similar, and that's your cerebellum or your hindbrain. Your cerebellum also helps us to control movement because what it does is it coordinates information from your cerebral motor cortex, from that primary motor area. It coordinates from your brainstem. It also pulls information from all the sensory receptors in your muscles, in your eyes, in your inner ear for equilibrium, in all of the proprioceptors or receptors in your joints that tell you where your muscles are in what position. So it helps us to use all that information to give precise timing and patterns to movements, coordinated movements, equilibrium, control even when you're not paying attention. So it helps us to plan movements as well. I intend to pick up my coffee. So coordinated balance, muscle timing, postural control, and even repeated motion. You can see that that can also become diseased as well. There is a disorder called cerebellar pathology or cerebellar disease, and one of the major indicators is what we call intention tremor. Intention tremors are tremors that are absent at rest. So again, if you rest your arm on the desk, it would be completely rested. There would be no movement. But when you decide to pick up your coffee cup, when you intend to pick up that coffee cup, that's when you see the tremors begin and even increase as you get closer to your cup. So you can't start and stop movements well. You may not be able to control that force either. You'll have poor posture and unstable gait. It's better if you have your eyes open. You may not sway, but when your eyes are closed with like the Romberg test, you really will sway. So signs of cerebellar disease: vertigo, kind of being dizzy, ataxia, kind of swaying. Your eyes may move left and right. That's nystagmus. The intention tremor is a big one. And there's also dysdiadochokinesia. That's an interesting word. Imagine putting your hand up. Go ahead and put your hand up so you're facing the palm of your hand. And now lay your other hand over that hand. Face the palm as well. Now, on the hand that's overlying, the one on top, switch it so that you face the back of the hand. So you're looking at your knuckles. Now face the palm, and the knuckles and the palm, and do it very quickly. That repeated motion would be impossible for you if you had dysdiadochokinesia. That rapid repeating, alternating movements are impaired in a person with cerebellar disease. So how do we get the information down through the spinal cord? So we're going to take a look at our descending pathways, or our projection fibers. We want to remember that this primary motor area uses large neurons called pyramidal neurons, and they have long pyramidal tracts that do not synapse. They cross over to the other side of the brain, or other side of the spinal cord, and then finally synapse at this anterior horn. You also have nuclei from the brainstem that will carry information of motor, again, down through the cord. But you can see they don't pass through the pyramids. They originate here in the brainstem. They're mostly concerned with, again, posture, control, balance, walking. But these guys, this particular set of neurons, they do not decussate. They do not cross over to the other side of the brain, and the innervation is not contralateral. So again, that would be from areas like your basal nuclei and your cerebellum. To finish up, we also have an area of the PNS that we're going to just briefly touch on, and that's the cranial nerves. Remember that our cranial nerves, 1 through 12 pairs of cranial nerves, have sometimes some that are just sensory, some are sensory and motor, and then some are just motor. Did you realize that 9 of them, 9 of the 12, are motor neurons that can deliver motor information to your head, your face, your neck, and even beyond that if you consider the vagus nerve. So I wanted to list out for you the cranial nerves that actually do help us to move. And I highlighted just those few that are sensory only. So the olfactory, the optic, and the vestibulocochlear are the only of the cranial nerves that have no motor skill at all. Well, thanks for listening to our motor audio, and I hope that that information was helpful for you, and please feel free to listen again.